Hey, good day class. So sa atong nga last part sa atong nga bacteriology nga topic. So this compilation sang halin sa first part which you have yung mga precautions, mga bacterial growth. So mas mas gin combine lang into one modules. This already 144 nga pages. So para ma-discuss naman ni, okay. We'll be skipping mga parts na nga na-discuss para mas makatipid kita sa aton nga time. So without further ado, so let now proceed sa aton nga module. So what is your bacteria? What are your bacteria? So bacteria are microscopic, single-celled organism that thrive in diverse environment. These organism can live in your soil, ocean, kag inside sa aton nga human guts. A human relationship with bacteria is complex. Sometimes bacteria lead as a helping hand, such as your curdling milk into a yogurt or helping with your digestion. In other cases, bacteria are so destructive that they also cause your disease like your pneumonia and also mga methicillin resistant nga Staphylococcus Aureus or your MRSA. So the structure of your bacteria is a singular, kung singular it is bacterium, are classified as your prokaryotes which are single-celled organism with a simple internal structure that lacks a nucleus and contains your DNA that's either floats freely or twisted like a thread of mass called your nucleus in us or in a separate. Your circular pieces are called your plasmids, and also you have your ribosomes at the that are spherical units of your bacterial cell where proteins are assembled from individual amino acid using information encoded sa ilang ribosomal RNA. So yung mga bacterial cells are generally surrounded into two productive coverings, your outer cell wall and ang iyang inner cell membrane. Then you have your certain bacteria like your mycoplasmas do not have cell wall at all, and some bacteria may have a third of your outermost protective layer called your capsules. Okay, ang ga-encase pagid. And you have your whip-like extension often covers your bacterial surfaces. The long ones are what you call your flagella and the short ones are what you call your villi. Kaya usually ang purpose atong nga flagella is for the motility and you have yung mga sex villi for transmissions ng ilang mga DNA and other such things. And you help bacteria to move around and also attach sa host. Okay, as simpler terms, your bacteria are single-celled organism which prokaryotic cell which are also single cell too that do not have organelles or true nucleus and are less complex than your eukaryotic cells. Bacteria with a capital B refers to a domain bacteria and others three domains of your life and the other two domains like such as your archaea, members of those mga single cell organism with your prokaryotic cells and your eukarya. Bacteria are extremely numerous and the total biomass of the bacteria on earth is more than all of the plants kag animals nga combined. Now, let us now proceed sa atong classification. Hey, classification, you have your gram stains in the is a test that used to identify your bacteria composition sa ilang cell wall. Name your names from your Hans Christian Gram, the one who developed the technique in 1884, and the test stain gram-positive bacteria or your bacteria that do not have an outer membrane and your gram-negative bacteria that don't pick up the stain. Yung colors natin ng gram-positive, simply lang the color for positive is blue or purple. Then for the gram-negative, it is either red or pink. For example, your Streptococcus pneumoniae or S. pneumoniae, which causes your pneumonia, is a gram-positive bacterium. But your Escherichia coli or your E. coli and also your Vibrio cholerae, which causes cholera, are your gram-negative nga bacteria. Now, sa aton nga mga three basic bacterial shape, you have your round bacteria, which is called your cocci. Then you have your mga cylindrical or capsule shape, or what you call your bacilli or bacillus, and your spiral, this is abruptly called your spirella or spirillum, kung singular. The shapes of the configurations of your bacteria are often reflected sa ilang name. For example, yung milk curdling Lactobacillus acidophilus are bacilli and your pneumoniae causing S. pneumoniae are the main chain of your cocci. Some bacteria takes other shapes as your stalker, square, and also stars. Now, let us proceed sa ilang reproduction. Like for the reproduction, most bacteria multiply in the process what you call your binary fission. Na-explain ko na that your bacteria will be split into two. According to your Cornell University College of Agriculture and Life Science, in this process, a single bacteria or cell, or what you call your parent, makes a copy of its DNA and grow larger by doubling its cellular content. The cell then split apart, pushing the duplicate material out and creating two identical daughter cells. Some bacteria species such as your cyanobacteria and firmicutes produced via budding. In this case, the daughter cells grew an offspring or offshoot of your parent, and it starts off a small grows until the same size as its parents, then splits off. So, anong tsura sa itong budding? Okay. You have here. Wait, hindi po ko lang sa internet. Okay, so your class is your budding. So you have here an example of a budding yeast. So usually sa microorganism, ang budding is usually sa mga yeast nga mga microorganism or yeast species. You have here one, which ga form sa ang bud, then ga dako ya size. Also ang mga buds mo, okay, can or ang offspring nga bud will make small portions or mga buds ang offspring pagid, so it will cause or dough branching. Tama na. So the DNA found in the parent or the offsprings are primary binary fissions budding. Are exactly the same. Therefore, bacteria cells introduce your variation into their genetic materials by integrating additional DNA, often from their surroundings, into their genome. This is known as your horizontal gene transfer. The resulting genetic variation ensures that bacteria can adapt and also survive as the environment changes or there are three ways of your transfer or aton nga horizontal gene transfer. First is your transmission and transduction kag conjugation. So, transformation is the most common process of your horizontal gene transfer and occurs when a short DNA fragments are exchanged between the donor kag ang recipient, and your transduction, which typically only occurs between closely related bacteria and requires a donor and recipient to transfer DNA by sharing cell surface. Then you have your conjugation. It requires the physical contact between the cell walls of your bacteria. The DNA transfers from the donor to your recipient through conjunction. A bacterial cell can transfer your DNA to a eukaryotic cell, or ang mga eukaryotic, di ba, is ang mga multi-celled layer, multi-celled organism, and your conjugation aids with the spread of antibiotic resistance nga genes. So you have your mga methicillin resistant nga Staphylococcus aureus. Then your bacteria in your human health and also disease. Bacteria can be classified into determining your human health, commensal or friendly. Okay, that is hatag sang benefit sa aton. Pareha sa aton nga lactobacillus nga para sa kita mo sa Yakult, and shares the space of the sources within our body and tend to be helpful. There are about 10 times more microbial cells than your body cells in our body. The highest number of your microbial species that are found in your gut according to your microbiologist. And the human gut is a comfortable setting for your bacteria with plenty of nutrients available for their sustenance. The 2014 review article published by the American Journal Gastroenterology, okay, is the study sa aton nga intestinal tract or sa aton nga digestive tract. The author mentioned that the gut bacteria and other microorganisms such as your as a helpful strain nga E. coli kag Streptococcus aids in your digestion and save off colonizing the harmful pathogens and helps the develop of your immune system. Moreover, in the disruption of gut bacteria has been linked to a certain disease condition. For instance, the patients with the Crohn's disease have an increased immune system response against gut bacteria sa iyang review 2003 nga published journal by the Lancet and other bacteria and cause infection. So you have your mga several bacteria ranging from your so-called group sa aton nga Streptococcus, you have your Clostridium perfringens or C. perfringens, E. coli ang imong Escherichia coli, and you have Staphylococcus aureus and can cause rare but very severe soft tissue infection called your necrotizing fasciitis, or what you call your flesh-eating bacteria. According to your CDC, or Center for Disease Control and Prevention, this infection affects the tissue surrounding muscles, nerves, and fat, kag blood vessels, and can also be treated, especially when caught being early. So you have here a computer-generated picture of your E. coli. So you can see ang iyang mga flagella, mga lagba, and mga pilaya ang mga gagmay or mga nipis. Now, sa aton nga antibiotic resistance, antibiotics are typically used to treat your bacterial infections. However, in recent years, improper usage of mga unnecessary antibiotic has prompted to spread several strains of your antibiotic resistance nga bacteria. In cases the antibiotic resistance, the infectious bacteria are no longer susceptible or hindi na pwede mapatay sa aton nga mga antibiotics. According to the CDC, at least 2 million people in the US are infected with an antibiotic resistant bacteria every week, leading to death at least for 2,300 at 23,000 people. Now, plenty much any infection that can think of as now is being infected and being associated with the some level of resistance. Okay, as infection disease physician and a hospital epidemiologist of the University of Washington Hospital and Madison Veterans Hospital, there are very few infections that we now treat where infections caused by a resistant bacteria is not a clinical concern. We have your MRSA, your methicillin-resistant Staphylococcus aureus, for example, is one of the most or more notorious antibiotic-resistant bacteria strains. It resists your methicillin and other antibiotics used to treat your Staphylococcus infections. They are acquired primarily through skin contact. Your MRSA infections occur in a healthcare setting such as your hospital and mga nursing homes, where it can lead into pneumonia and also bloodstream infections or sepsis. Your MRSA also spreads in the community, especially in a situation where there is a lot of skin exposure and other physical contacts and the use of a shared equipment. For example, among these athletes, ang mga tattooan, okay, tattoo parlor, and in the daycare facilities and also school. Your community-acquired MRSA often causes skin infections. A very important facet combination antibiotic resistance is very to be careful about their use. It is important for us to use antibiotics intelligently. Okay? Hambal ng isang cringe to life science, and you only want to use an antibiotic when you have to clear cut bacterial infection. That's why it is very important nga if you are given with an antibiotic nga treatment, you need to finish it from day 1 to the last day. Dapat wala ka sang may i-miss. Okay? Now, for your bacterial growth here, class, so you have bacteria that can replicate approximately every 20 minutes by your binary fusion, which is simple form of cell division. This level of replication depends on the availability of your nutrients and other sustainable conditions such as yung mga temperature, mga pH, and so on and so forth. You have your one bacterium, isa kabilog na bacteria, in 20 minutes, magiging 2 na siya. In 40 minutes, magiging 4. Ang imo nga 4 nga bacteria in 60 minutes will turn into 8 nga bacteria. So, binary fusion lang siya. So, there are many ways to grow or culture bacteria. So, aton nga na-discuss, you have your broth kag aton nga agars. So, nutrient broth is solution of liquid siya. Culture medium allows liquid gel or provide the nutrients needed for bacteria to grow successfully. This must include carbohydrates for your energy and nitrogen for protein synthesis plus other minerals. Your agar plates naman yung class are created by pouring a hot molten agar, though gelatin-like na, into a sterile petri dish, which then allows to set. Your bacteria can be spread into your plates and allow them to form mga individual colonies for a specific bacterium or for identification. You have mga uncontaminated nga cultures is a specific bacterium that is going to be cultured. Another other contaminating bacteria would compete for nutrients in broth or agar. Some bacteria would be harmful, such as your pathogens, and would complicate the results of your experiment when testing the efficiency of your antibiotics or other antimicrobial compound. Now, use of aseptic technique. Okay, so hindi ko na niyong ipagliwaton. This was already discussed na. Okay. Okay, the use of aton nga Bunsen burner, the pag-flame sa imong inoculating loop, proper opening, closing sang imong petri dish kung na-inoculate mo na. Then you have your colonies of your bacteria. Bacteria are microorganisms and also individual that cannot be seen without the help of our microscope. However, when grown in an agar or petri dish, each individual cells divide multiply times to form your visible nga colonies. You have here ng colonies. If we count the numbers of individual colonies on the bacteria and the plates, it is possible to estimate the number of individual bacteria in our original sample. In order to make the accurate calculations of the number of your bacteria in a sample, the original sample will need to be diluted. Is what you call your serial dilution, which only when the sample is sufficiently dilute and can provide clear individual colonies and calculations be made. So you have here, wala na dilute, so it is a pure. Ay, nagkuha siya around 1% or 10%. And, okay, by 10% na lang ginkuha yan di. Then, move 10% pagid, 10% naman sa halindi, and 10% halindi, and 10% pagid. So, therefore, it is diluted. So, gagamay ang mga pwede mo ma-isolate na bacteria. And visible, mas clear kaysa nga dili is damo nga colonies ang bacteria. Then you have your bacterial growth. Bacteria produce at regular intervals. For example, every 20 minutes, they allow the mathematical calculations to be made in order to predict how many bacteria will be present in a given time. So how to calculate the number of your bacteria in a population? Okay, for example, a mean division time for your bacterial population is around 20 minutes. If the observation begins 1 bacterium, calculate how many bacteria will present in your 6 hours. So, pila ka 20 minutes sa 6 hours. Now, in order to answer this, you need to split the calculation into 2 sections. Calculate how many times the bacteria divides in your 6 hours. Okay, in this sample, the bacteria divides in every 20 minutes and will therefore divide 3 times in every hour. So, you have here 60 and 20 minutes. So, katatlo. Divide isa mo. Wala na nabatang sinayin. Okay, if the bacteria grows for 6 hours, okay, each bacterium will divide 3 times per hour. So, it is 3 times 6. So, you have 18 times. Calculate the number of your bacteria in your populations. Every time the bacteria reproduce, the number doubles. To calculate the number of your bacteria at every end of your growth period, you can use this equation. Bacteria at the beginning, so isa, and the growth period, which is kadua, di ba, kay binary fusion sa kag ang iya nga exponent kung kapila siya nag-raise. So, the number of bacteria isa, then number of division, you have 18, di ba? Because it is 20 minutes in 1 hour, katatlo siya mag-multiply. So, you have 6 hours in every hour katatlo. So, 3 times 6, so it is 18 times. So, you have 1 times 2 raised to 18. So, man nakasimple naton nga estimation sa aton nga bacterial growth. So, for higher mark, you should express answer in your standard form. For example, if the answer is above 264,000, 144 bacteria can also read and your scientific notation, which is 2 times 6, 2.62 times 10 raised to 5. So, kabalo mo ko mag scientific notation since nagyan nila sa ato nga chemistry. Now, following your bacterial growth, as shown and above, the experimental growth for a bacteria can give a large number of bacterial cells in short period of your time. The important is where the food storage is being concerned and we are able to slow its process to prevent spoilage. The rate of each bacteria divide can be produced or reduction sa ating temperature, keeping the food in your refrigerator slows your bacterial growth. Food can be kept in a longer before spoiling. In a frozen, if the food is frozen, the bacterial growth stops. However, the bacteria that are not being killed. So, in increase aton nga temperature would decrease your bacterial growth. Cooking food at very high temperature will kill your bacteria. So, your types of bacteria. Ay, na-discuss na naman ni. Hindi na napagliliwa to. You have your shapes. Okay, ang round ta, you have your coccus. Okay, rod-like are your bacillus. Your comma are your vibrio. Your spiral are your spirella or spirochetes. Yung mga cell wall composition, gram-positive, take peptoglycan layer, gram-negative, lipopolysaccharide nga layer. Then you have your gaseous requirements. Anaerobic, okay, it's either obligate or facultative. Then you have aerobic and anaerobic. Now, sa itong mga nutrients pattern, you have autotrophic. Autotrophic are either photosynthetic or chemostatic or heterotrophic. They produce their own nutrients. Then, you have bacterial subclassification. You have your diplo. It's a prefix na gina-add mo sa ilang name. So, diplo. Big sabihin, they are in pairs. Your cocobacilli. Okay, the combination sa mga coccus and your bacilli nga rod. So, it's the oval. Ilang shape. Vibrio, yung latura. You have Helicobacter, hexical. Then you have your Spero, Strepto. Strepto is, ibig sabihin niya na in chains. You have your Streptobacilli. They are bacilli that are in chain. You have your Spirella. They are coiled. You have Cornybacter. Okay, do club, do baton, or iya ka badminton. Then you have your Stapi. Okay, Staphylo. Ibig sabihin na in clusters. That's why you have a Staphylococcus Aureus. So they are a cluster of cocci. Okay, and ang Aureus is the bacteria. So Staphylococcus Aureus. No, di mo makuha ilang mga name. You have your Mycobacteria. You have your Spirochetes, spiral. And you have your Streptomyces. Now, your prokaryotes are organisms whose cell lacks a nucleus, pro, before, and karyon, ibig sabihin niya na nucleus. There belongs to the kingdom Monera, and they have a further classification of two distinct domain, your Actinobacter, found in the extreme environment, like your high temperature and also high concentration sa aton nga salt or pH. You are what you call your extremophiles. Then you have your Eubacteria, traditional bacteria including most of the pathogen, for example, your E. coli and Staphylococcus aureus. Mga features sa ating prokaryotic cells, na-discuss na naman isang ligad. Kindly, scan lang. Then you have applications, binary fission. Okay, already discuss ko na discussion. Yung shapes, liwat, cocci, spirella, bacilli, ang common. Then you have your cell wall. Okay, gram positive kang gram negative. Gram positive, color ya. Tandaan nyo lang, mas thick ang imong gram positive, it is color blue. And gram negative is quite thinner and it is like lipopolysaccharide. Then, growth gram negative cell walls contain your peptoglycan, also known as your morium. Okay, tandaan nyo lang na mga naka-highlight. Then, you have your structure. And discuss ko naman dito na pagduwaton. Okay, with some more specific discussion sa aton nga cell wall, pero na-discuss ko naman previously. Okay. So, this is 44 pages ni nga daan ka. So, this is a compilation gid ni sang tanan nga halin sa introduction sa aton nga microbiology, because microbiology is do intro mo giniya sa bacteriology. Then you have your mga oxygen requirements or aton nga physical requirements sa bacteria. You have your oxygen. So you have here ang aton nga anaerobic, whereas one of the most prominent difference between, practice ma-clear sa inyo, the difference between your bacteria is the requirement for and response to atmospheric oxygen. Whereas, essentially, all eukaryotic organism requires oxygen to thrive. Many species of your bacteria can grow under anaerobic condition. Okay, ano ng anaerobic condition? Okay, so may mga blue na it is being linked in a definition. Sabi yung mga Britannica sources. If you have internet connection, you can open it in link. So you have anaerobic conditions. Bacteria that requires oxygen to grow are called your obligate aerobic. Ibig sabihin, if my name nga obligate, okay, they are 100% required to have oxygen to grow. Kapag hambal nga anaerobic, ibig sabihin ng okay, they are not required an oxygen. Then you have these bacteria require oxygen to grow because their methods of energy production transfer of electrons sa oxygen, which is the final electron acceptor for electron transport reaction. Your obligate aerobes, okay, includes your Bacillus subtilis, your Pseudomonas aeruginosa, your Mycobacterium tuberculosis, your Acidotheobacillus feroxodecans. You have bacteria that can grow in the absence of your oxygen. You have your Clostridium, then your bactericides, and methane-producing archaea. They are called your obligate anaerobe. Ibig sabihin, kung may obligate, ganun sila nga name, they have 100% nga requires zero oxygen gid para mag-grow. Kaya kung may presence of oxygen, they will die. Okay, because their energy generating metabolism process are not coupled with consumption of oxygen. In fact, the presence of oxygen actually poisons some of their key enzyme, and it will kill the bacteria. Now, your facultative aerobes naman yung class can change their metabolic process depending on the presence of oxygen, using the more efficient processes of your respiration and presence of oxygen in a less efficient processes are your fermentations in the absence of oxygen. For example, your facultative aerobes like your E. coli and Staphylococcus aureus. Your facultative anaerobes, okay, they will grow in either with or without oxygen. Muna ka, simple. Then, you have your mga catalase. Explain to naman na. Your temperature. Okay, temperature sa bacteria or ang ilang preferred optimal temperatures you have here. Your bacteria can grow at the temperature of less than about 15 degrees Celsius or 59 Fahrenheit are your Phycrophiles. Okay, Phycrophiles, tandaan nyo na, Phycrophiles, low temperature, the ability to grow at a low temperature are unexpected. Since the average subsurface temperature of soil is around temperature sa ating 12 degrees Celsius and 54 degrees Fahrenheit, and 90% the ocean temperature is around 5 degrees Celsius or 41 degrees Fahrenheit or colder. Now, your obligate phycrophiles, which have been isolated from Antarctic Ocean waters and sediments, have optimum growth temperature around 10 degrees Celsius and do not live or survive if exposed around higher temperature pa da. The majority of your phycophilic bacteria are your gram-negative genera Pseudomonas, Flavibacterium, Actinobacterium, Alcagenis, and Mesophilic. Ang mga mesophilic naman yan, lain naman yan. Okay, mesophilic nga bacteria are those with the optimum growth around 20 to 45 degrees Celsius. So, ang body temperature sang human is nabilong sa mga mesophilic bacteria, which is ang atong normal body temp is 36.5, di ba? So, ang mga bacteria nga mabuhay sa aton are mesophilic bacteria. Okay, although they survive and grow in the temperatures between 10 to 15 degrees Celsius, and animal pathogens are mesophiles. Now, for thermobacterial prokaryote, can grow at the temperature higher than 60 degrees Celsius. These temperatures are encountered in the rotting compost piles, hot springs, mga ocean nga mga geothermal nga vents, okay, man near mga ocean volcano, na runoff or mga hot spring. Thermophiles, okay, thermophiles are mga higher temperatures around 60 degrees higher or around 140 degrees Fahrenheit. Muna tatlo sa aton nga temperature. Sa aton nga pH, okay, most bacteria grows in the neutral pH value around 5 to 8, although some species have adapted to the life of more acidic or alkaline nga extremes. An example for acidophilic nga mga bacterium are your ferrooxidants, when coal seems to be exposed in the air, mining exploration, ferritic ferrous sulfide deposits are attacked by ferrooxidants to generate sulfuric acids, which lowers your pH to 2 or even 0.7. So it is acidic. However, the acid tolerance for sulfurans applies only to sulfuric acid, since these bacteria die when exposed to equivalent concentration of other acids, such as your hydrochloric acids. And many bacteria cannot tolerate your acidic environment, especially under anaerobic condition, and a result, a plant polymer degrades slowly in an acid pH between 3.7 and 5.5. Then you have, in contrast to your acidophilic bacteria, your alkalophilic nga bacteria, alkalophilic. Okay, ang ilang preferred nga pH condition is 10 to 11. Acidophiles or acidophilic have been isolated from your soil, and most are species from your gram-positive nga genus Bacillus. Okay, so hindi na nga padgayan gid. Okay, mga itophilic bacteria. Scan nyo lang na class, hindi ka na gid man din pagpadaluman gid. Kaya mga saprophytes, okay, they are major decomposers in your nature, so obtain their food by detritus and dead organisms. Saprophytes. Okay, they are symbiotes. They live in symbiotic association with other plants and mutual benefits. And some species of your Rhizobium or are Rhizobium leguminosarum live in the roots of your nodule of leguminous plants, forming an intimate symbiotic relationship. Bacteria fix atmosphere nitrogen for your plants, and in return, it gets protection. Your Escherichia coli live in human intestines as commensal. It feeds on undigested matter, checks the growth or sa aton mga putrefying your bacteria and produce your vitamin B and K. And the presence of your E. coli in water supply indicates contaminations by sewage. Kaya usually atong E. coli ginapagbuhat naman na sa aton nga aton nga as waste sa aton nga tae. Then you have your mga autotrophic bacteria or autotrophs. Okay. Dili na your photoautotropic or photosynthetic, which mix use of light energy, and your chemotopic or utilize chemical energy. You have here mas differ nga or explanation sa ating phototropic bacteria and your chemotropic. Hindi ka nag-idvag padalman because I already explained it mga previous mga chapter. So what is a normal flora and why it is important? So before pa lang nga daan sa ating microbiology, I already discussed what is your normal flora. It is very simple. These are bacteria that are found normally nasa imo nga lawas or kung hindi man sa imo nga environment that doesn't cause any harm sa imo as a person kag wala ga cause damage sa environment or kung sa diin man sila ga attach. That is what your normal flora is. And so why is it important? When ako gabasa, why it is important? Because it takes the nutrients away from mga pathologic nga mga microorganism nga possible mag-grow sa area or sa imo. Okay, that's the importance of your normal flora. That's why you don't need to overly be cautious sa imo nga self nga tama kagid ka clean nga dapat 0% way kagid sa bacteria, because you will be killing your normal flora, and in case nga madapuan ka sang pathologic, therefore you will get sick instantly because you don't have normal flora that consumes the nutrients. Ang tanang nga nutrients nga sa lawas mo will be given to your pathologic. Therefore, mas dasig sa mag-proliferate and you can't do anything about it. So, you have bacterial flora sa aton nga community, bacterial flora sa hospital and long-term care facility. So, maari mga pathologic na because they are ara sa hospital kag mga healthcare nga facility. Mga pula, okay, hindi ko na pagpalawigon gid. So, kamo ni bahala scan. So, these are mga normal flora. This is very helpful sa atin lawas. Then, you have Staphylococcus Aureus. Okay, mga common bacteria that you need to know. Okay, Staphylococcus Aureus. Mga Staphylococcus species, your Streptococcus species. Mas-stop lang or ma-discuss lang ko if there are mga important types. So, from the name niya nga stop gani, a stop means a cluster. Coccus means mga circles or spears. So, they are a cluster of spears. They are gram-positive. So, depende na lang nasa iglaman nga species. Now, your strep, ibig sabihin niya in chain. Okay, strepto in chain, and your coccus means like no. Okay. Langgit. Okay na. In chains, then, amo nila tsura class. Chains. Katirin-tirin. Na mga ilang species, mga host, of Streptococcus pyogenes. Then, you have mga differential. Okay, this is very important. Why is it? Di ba nag-discuss na to about agars? You have your blood agar plate. So, this is a blood agar plate. Okay? This is inoculated with a strep pyogenes or different type of a streptococcus. Okay? You have here a gamma reaction. Okay, a gamma reaction is there is no change sa aton nga plate. May grow lang sang colony but there is no change. Okay? Ibig sabihin the Streptococcus species uses the enzyme to break down sa aton nga blood and produces a clear area sa aton nga surrounding. Your no effect, you have your alpha, incomplete hemolysis that produces a greenish discoloration about sa colony. So this is your alpha, your beta, and your gamma. So duwa ka gamma. Okay, although diri you may see do may puti-puti but it is not an beta. Okay, a beta is a enzymes are being lysed sa aton nga red blood cell and completely. Okay, ibig ano na meaning niya sang lyse, okay? An enzyme produced by aton nga bacteria, okay, lyses. Okay, anong ibig sabihin sang lyse gani? Lyse is gina-destroy aton red blood cell, therefore completely clearing your agar plate. So amo niya tsura. So it is clear sa surroundings ang imo nga colony. So amo ni mga black spot that are your colonies sa ating mga bacteria. Ang dire it is a incomplete hemolysis, therefore it is color green sa palibot sang colony. So that is very simple. You have here what? Gamma hemolysis, wala sang natabo. Your alpha produces a green incomplete lysis. Then you have your beta is a complete lysis sang red blood cell. Okay, gin-consume gid sa sang bacteria. Your lactobacillus species, you have here usually sa mga rod shape lactobacillus, either rod shape non-spore forming bacteria, either gram positive, aerotolerant, or anaerobes, and microaerophilic. So dito namin nga part na yan. So dito pa. Okay, so in humans, they constitutively are a significant component of your microbiota at the number of body sites, such as your digestive tract and urinary tract, kag genital system in women of European ancestry. Your lactobacillus species are normally a major part of your vaginal microbiota. Your lactobacillus forms in the biofilm in your vagina and gut microbiota, allowing them to persist during harsh environment condition and maintain ample population. So, hindi ko gid ni malimtan ang lactobacillus because when I was reviewing for my board exam, din ask me what is the most common bacteria in a vagina. So, nakibot ko nga ang answer is lactobacillus. Because sabi ko lactobacillus is usually sa amuna sa Yakult before related sa stomach but it is ammonia ang pinaka main nga bacteria sa vagina. Okay, so the female vaginal tract is one of the principal colonization site for your human biota, and there is interest in the relationship between the composition of your bacteria and human health, with the domination by a single species being correlated with the genital warfare and a good outcome for your pregnancy. It is around 70% of women. A lactobacillus species is dominant, although it has been found to be vary between American women of European origin and African origin, and later groups tending to have more diverse vaginal microbiota. Kaya askin ginasadihan man ang normal flora sang vagina kag ang ilang race. So hindi man sa pagiging races. Kanya probiotics. Okay, lactobacillus species administered in the combination with other probiotics benefits in cases the irritable bowel syndrome or IBS. Although the extent of efficiency is still uncertain, the probiotics help treat IBS or irritable bowel syndrome by returning the hemostasis of your gut microbiota. Experience usual high levels of opportunistic bacteria. In addition to your lactobacillus species can be administered as probiotic during the cases of infection by an ulcer-causing bacterium like your Helicobacter pylori. Your Helicobacter pylori is linked to a cancer, and antibiotic resistance impedes the success of current antibiotic-based eradication treatment. When lactobacillus probiotics are being administered along with the treatment as an adjuvant, its efficiency is substantially increased and side effects are being lessened. Also, your lactobacillus is used to help control urogenital and vaginal infections such as your bacterial vaginosis or BV. Lactobacillus produce a bacteriocin to suppress the pathogen growth of a certain bacteria as well as lactic acid same sa aton nga hydrogen peroxide. Lactic acid lowers the vaginal pH to around 4.5 or less, hampering the survival of other bacteria, and ang imo nga hydrogen peroxide establishes the normal bacterial microbiota and normal vaginal pH. In children, lactobacillus strains such as yung Lactobacillus ramonusus are associated with the reduction for your optic ischemia as well as mga dermatitis due to anti-inflammatory cytokinesis secretes by your probiotic bacteria in addition to your lactobacillus in order. Other probiotics organisms and ripened milks like your yogurt aids develop immunity to mucous intestine in human by raising the number of your LGA. So oral health, hindi na mag-discuss again. Your food production, hindi ko mapadalo. Same lang na ihapon. Okay? This is already summary na sa aton nga microbiology sa aton nga bacteriology nga topic. So iskan nyo na lang ni class sa aton nga last part because at least maka-next pa kita nga topic. So that would be all sa aton nga bacteriology nga topic. So medyo madamo ni pero pwede nyo man din daan magamit ang search nga function diri sa aton nga PDF. Okay, search mo lang. Okay, if we're looking for about Aureus. So it will search kung ano nga topic ang video mo search din. Because this already it is very helpful to specify search nga tool. Okay, kaya link sa first nga part na discuss na ni. So do mas compact lang. Although may mga last nga topic, pero mas nakafocus siya sa mga bacteria. So that will be all. Stay safe always, class, and God bless you.